Part 1. Personal Safety Precautions Level 3. In non-contaminated area. Put on the cap. Then N95 compliant mask. Check if the sealing is good. Then the disposable protective clothing. Then the shoe cover. Then the waterproof shoe cover. Then the first layer of latex gloves. Check if the sealing is good. Crouch to test if the clothing fits well. Put on the second layer of protective clothing if available. It is necessary to ask your colleague to help and check. Put on the protective goggles. Then the second layer of latex gloves. Check if the sealing is good. After dressing up, pass the buffer room or corridor to go to the work area as soon as possible. Part 2. Deactivation of the sample. 2 persons are required to perform the NAT test in the laboratory. Keep disinfectant alcohol and absorbent cotton in the biosafety cabinet. Use absorbent cloth to handle accidental sample spillage. Open the secondary container of the sample. Spray alcohol to sterilize the outside of the primary sealing package of the sample. Check if the sealing of the primary sample package is intact before inactivation procedure. Inactivate the sample at 56 centigrade Celsius for 30 minutes with water bath, dry bath, or other proper heating devices. After 30 minutes, take out the sample package, wipe it with alcohol. Take it back to the biosafety cabinet. Part 3. Manual nucleic acid extraction. The reagent preparation should be performed by another person in a separate room. Step 1. Prepare wash buffer AW1, AW2, and carrier RNA. Add 130 ml dehydrated alcohol to the wash buffer AW1. Add 160 ml dehydrated alcohol to wash buffer AW2. Label the container immediately and clearly. Add 310 microliter AVE in 310 microgram carrier RNA. Invert it to mix well. Wash buffer AW1, AW2, and carrier RNA are ready. Transfer the preparation through interlock clean room path, or send them by another person to the nucleic acid extraction. 2. Cell lysis. First, get sterilized 1.5 ml EP tubes. Dispense 560 microliter AVL lysis buffer. Add 5.6 microliter carrier RNA preparations into it. Add 140 microliter inactivated sample. Vortex mixing for 15 seconds. Room temperature lysis for 10 minutes. Short spin the tubes to collect the lysis product at the bottom and avoid possible contamination of the remnants on the tube cap. Add 560 microliter dehydrated alcohol after lysis. Vortex mixing for 15 seconds. Short spin the tubes again to avoid contamination. Add 630 microliter lysate to the spin column. Centrifuge at 8,000 rpm for 1 minute. If the centrifuge is outside the cabinet, sterilize the external surface of the tubes whenever you take them in or out the cabinet. Pick up a new tube for waste collection. Take the tube out of the centrifuge with care. Pick the spin column out and put it into the new waste collection tube. Add the rest of the lysates, about 630 microliters, to the spin column. If the sample is more than 140 microliter, repeat the procedure until all the lysates are processed with the spin column. Centrifuge at 8,000 rpm for 1 minute. Transfer the spin column to a new tube with care. Add 500 microliter AW1 to the spin column. Important: dispense slowly with the tip in contact with the inner wall of the spin column. Centrifuge at 8,000 rpm for 1 minute. Transfer the spin column to a new tube with care. Add 500 microliter AW2 to the spin column. Centrifuge at 14,000 rpm for 3 minutes. Transfer the spin column to a new tube with care. Top speed centrifuge for 1 minute to ensure no AW2 left in the spin column. Pick a new sterilized 1.5 ml EP tube to collect nucleic acid elutes. Transfer the spin column to the tube with care. Add 40 to 60 microliter AVE to the spin column. Important: dispense the elution buffer evenly across the whole surface of the spin column filter. Close the cap of the EP tube with the spin column inside. Cut off the cap of the spin column itself. Room temperature incubation for 1 minute. Centrifuge at 8000 rpm for 1 minute to elute the nucleic acid. Discard the spin column. Keep the nucleic acid extract in the EP tube and label the tube. Part 4. Automatic nucleic acid extraction. Follow the kit menu to get the reagent preparation ready. Add 200 microliter inactivated sample. Follow the instrument menu to run the extraction. Recover the nucleic acid extract after the instrument stops. Part 5. PCR mix preparation. The PCR mix should be prepared in a separate room by another person. Prepare the reaction mix proportional to total test numbers. Vortex the mix with MXS and short spin with D1008 palm centrifuge. Send the mix through the interlock cleanroom pass to the separated sample handling room. Dispense 20 microliter mix for each reaction. Add 5 microliter sample RNA extract, or negative or positive control. Important: 1 positive and 3 negative controls for each batch. The negative controls should be placed randomly. Seal the PCR strip. We recommend to add PCR templates within a separate biosafety cabinet. Send the PCR strips through the interlock clean room pass or by another person to the separate PCR amplification room. Part 6. qPCR analysis. The qPCR should be performed by another person in a separate room. Fetch the PCR strip from the interlock cleanroom pass. Short spin the strip with D1008. Follow the kit menu to set up the qPCR protocol and run tests. Read the kit manual carefully before data analysis. Ensure that you understand the sensitivity and other limitations of the kit to interpret the test data properly. Ensure that the test results meet the QC criteria. Keep the used PCR strip closed. Seal the strips with plastic packs and discard them immediately into the dedicated garbage bin. Part 7. Post-test disinfection. After tests, spray 75% alcohol to disinfect the gloves. Discard the first layer of the gloves into the garbage bin inside the cabinet. Wipe the benchtop and the floor with 0.5 to 1% chlorine disinfectant or 75% alcohol. Seal the medical waste with triple-layer garbage bags. Put the bag inside the dedicated garbage bin for novel coronavirus tests. Label the bag with information for waste sorting. Turn on the ultraviolet light to disinfect the cabinet and the room for 30 minutes. Spray 0.5 to 1% chlorine disinfectant or 75% alcohol evenly on the protective clothing with the help of other colleagues. Take off the clothing and discard them into the dedicated garbage bin. Follow the specific sequence to take off personal protective wearings in the buffer room or corridor after you leave level 2 areas. Put on another pair of secondary gloves. Take off the goggles and immerse them in 75% alcohol. Take off the primary protective clothing from neck to toe, from inside to outside, and wrap the outside with the inside. Take off the secondary gloves. Then the mask. Then the cap. Then the shoe cover. Then the first layer of latex gloves. All the waste including disposed clothing should be sterilized with autoclave. Strictly distinguish the waste before or after autoclave. Sterilize the waste at 121 centigrade Celsius for 30 minutes with autoclave. Check the color of the autoclave indicator tape to confirm the effectiveness. Handle the waste as general medical waste thereafter.